Hi, I'm Dr. Drew Bogner, President of Molloy College, and I'd like to welcome you to the Public Square 2.0. In this program, we will be bringing you some of the great faculty that currently teach at Molloy College. They will share their insights on subjects that we hope you will find both entertaining and enlightening. Today's speaker is Jodi Evans. Jodi is an alumna of Molloy College and also an associate professor of biology in the Division of Natural Sciences. She was a driving force behind the development of the research series of courses in the Department of Biology, and she continues to coordinate the research program. Jodi is an active biomedical research scientist with a particular interest in adult stem cell research. Please join me in welcoming Jodi Evans. Jodi, welcome.
Thank you so much, Dr. Bogner, for inviting me to speak today, and thank all of you for joining us today. So many things might seem futuristic about medicine today, and certainly using adult stem cells to treat disease may sound like one of them. But I'm here to talk about this topic today and let you know how much closer this really is and how you need to stay up to date and informed in this ever-changing world of medicine. So first, I'm going to start with what is an adult stem cell. So I almost said mesenchymal stem cell because in my world, these are called mesenchymal stem cells. But so for our purposes today, we're going to say adult stem cell. So what is an adult stem cell? An adult stem cell is a cell that is present in most tissues of the adult. But our major sources that we're investigating today are from the bone marrow and from fat tissue. What makes these cells very important today, or what adult stem cells are so important and what makes them unique, is that they are from the adult, and that is very unique in that the ethical concerns are very nominal. All right, so what makes these candidates? What makes these candidates for treatment of disease? What are the characteristics of adult stem cells that make them efficient in treating disease? So these major characteristics are, first, they're able to differentiate, or they're able to become many different cell types. This means that they are a precursor cell, and if they're grown under the right conditions, these cells can become bone cells, they can become cartilage cells, they can become nervous cells. It's fascinating. It's really fascinating. It's amazing that when we grow them under the right conditions, they can become these many different cell types. And this is what makes them efficient in repair. A second characteristic that makes them able to be used for treatment of disease is that they are able to regulate immune cells or immune cell responses. They are also able to escape the host immune system. And so what does that mean, escape the host immune system? They are cloaked from the host immune system. All of our cells in our body have a tag, and our tags are unique among individuals. They're unique. So this allows our immune cells to identify our own cells as self. Well, these cells don't have that tag, and this is what makes them useful and in treating other people. So meaning that they could be donated from one person to another, and they won't be identified as a foreign cell if they are donated. So just to give you some examples of how they're used in repair, as we talked about, they can become different types of cells. So we're using them to treat diseases that involve degeneration or damage. And one of these diseases is osteoarthritis. So osteoarthritis is a disease where there is degeneration of the cartilage that protects our joints, the cartilage between bones that reduces friction and allows cushion. And when this degenerates, this is painful, and you have bone-on-bone inflammation and pain. But because adult stem cells can become cartilage cells, we can use them to repair this cartilage damage. Similarly, with heart attack, we have damaged cardiac tissue or heart tissue. These cells can also be used in the same way to repair this damage after heart attack. Another example of how they're used in repair is in peripheral artery disease. Maybe you don't know what peripheral artery disease is. It's a complication, a major complication or a common complication of diabetes. What happens with diabetic patients is their peripheral vasculature, let's say for example in their calf muscle, it becomes clogged and inefficient, and these cells are able to, because they're able to form many different cell types, they are able to create new vascular networks in these tissues to aid in repair. To give you some examples of how they regulate, how they're used, how the regulation of immune cells are used to treat disease, we use them to treat autoimmune disease like rheumatoid arthritis. So autoimmune disease, let's define it. What is an autoimmune disease? Auto, meaning self immunity. So what happens with an autoimmune disease is the person's own immune cells target that person's tissues, and it results in an inflammatory, damaging process. Well, these adult stem cells are capable of interacting with those immune cells and suppressing those damaging inflammatory processes. In addition to rheumatoid arthritis, they are used to prevent transplant rejection. In the medical world, we call this graft versus host disease. I'm sure you've all heard that when somebody receives a transplant, there is times where there is a rejection. And this is that person's immune system not recognizing that heart or organ as self, and that immune system mounts an attack against that organ. Well, these adult stem cells can be used to suppress that reaction and resist that rejection of the transplant. So through all of this, I keep mentioning this inflammation. This is a primary focus of my work, is how these adult stem cells control or can interact with immune cells and control an inflammatory response. So let's talk about what is an inflammatory response. An inflammatory response is a very necessary response. It's a major component of our immune defense. So it's good. However, when we have inflammation that goes unregulated, it's damaging. So I study with my students, I study how these adult stem cells interact with a particular immune cell. And I like to call this immune cell the Pac-Man of immune cells in the scientific world. This is called the macrophage cell. And this cell, I call it Pac-Man because it is responsible for engulfing damaged tissue. It's responsible for engulfing and getting rid of microbes. So therefore, Pac-Man. But this cell is also very active in the inflammatory response. And early in inflammation, this cell is pro-inflammatory. It needs to be. We need that initial inflammation to control the problem. But it has another face. Not only is it pro-inflammatory early, it turns and becomes anti-inflammatory later and is active in repair and resolution of inflammation. And these adult stem cells can help to support that early inflammation and can also support and influence, I guess you could say, that transition into the reparative, restorative cell. So I keep talking about adult stem cells and where we get them from. We get them from the tissues. But not all adult stem cells are the same. And we take them from the tissue, we have to separate out the stem cells from the other tissue cells. So we isolate them using specific markers on the cell that identifies them as stem cells. So an active area of investigation right now, and one that I've been doing with my students in the laboratory, is looking at the relationships between these markers and their function. So we can identify specific sets of cells that would be support inflammation, and specific cells that would then maybe suppress inflammation. So that's a very active area of research. So how close are we, you might be asking? How close are we to using these in the clinic, in the mainstream? We are closer than you may think. As of 2015, there were 493 clinical trials worldwide investigating the efficacy of these cells in the treatment of the diseases that I've discussed with you today. So we are much closer than you might think. So what does this research mean to you? There are promising new treatments on the horizon for these age-related degenerative diseases. And so what your job is to stay informed, stay up to date for the sake of your health and the health of your families. Thank you very much.
Welcome back to the Public Square 2.0. I'm Drew Bogner, president of Molloy College, and I'm here with Jodi Evans, associate professor of biology at Molloy College, and she's been talking about the promise of adult stem cells and how they can be used for the treatment of disease. So Jodi, I really appreciated your conversation. So we've talked about some of these things. I used to teach biology, so I kind of have an idea of how some of this works. So stem cells are really unique and that they have the capacity to be shaped and become different kinds of cells and to continue to regenerate, correct?
Mm-hmm. Yes.
What happens to that's really special, but the reason it's special is because our other cells as adults, many of our other cells don't have that ability, right?
You're absolutely right. Yeah, they don't have that ability. These cells are the precursors to those cells. And so they still exist, even though the other cells have differentiated to become their mature cell, they're going to stay that way. But we have some remaining in our adults' tissues that we have discovered, and now we're discovering ways to identify those.
Right, and I think that's special. So like if I damage the cartilage in my knee, well, that cartilage, those cells in that cartilage don't really have the ability to do much in the way of regenerating, correct?
No, cartilage is actually one of the tissues that has the least amount of ability to repair. It's not vascularized.
Right.
It really does not have a big ability to repair itself.
So I can go in and do a knee replacement and put in some artificial kind of joint, but there's not much else we can do. Probably there's some other techniques, I suppose. So with adult stem cell, though, if you can get it to act like cartilage,
yes,
then that's how you solve the trick. So that's one of my questions is, so I isolate these cells, the adult stem cells that have still this latent ability to differentiate. How do I get it to act like cartilage in my knee and not like a skin tissue or blood or bone marrow or something else? How do you get that to happen?
So we give them environmental clues. So we give them environmental clues. We take them out of the body and we can grow them under specific conditions in culture, and then we can prepare them to become cartilage cells when they're used. We're also, as I was explaining, looking for different markers on their surfaces. So we can identify them through an array of markers that then will predict which pathway they will take and become, whether they would become a bone cell or a cartilage cell.
So you're saying those markers would tell you if they can become those cells, or whether they can be more effective in becoming those cells?
A little bit of both.
Yeah.
Yeah, a little bit of both.
Yeah. So and the exciting thing about this is, if we went back 10 years ago or 15 years ago, people knew that the stem cells had potential to differentiate into other tissues, but they really weren't thinking that adults had these stem cells
No, not as much as they have today.
today.
But
So that's a significant ethical change for us, because the main source of stem cells previously was embryos and things of that nature before we started identifying stem cells in adult tissue. And I think that's a significant advantage to us ethically, to be able to realize that we can do this
absolutely.
and find those cells in a much more ethical manner.
And it's been explored. It's exponential. If you look at the literature on the type of stem cells that I'm talking about, the adult stem cells or the mesenchymal stem cells, if you look at the literature over those years, it has just skyrocketed and continues to grow, even 25% over just the last year alone.
I remember talking to Father Tom Hartman about this, I don't know, maybe a decade ago, and he was so excited that they'd found promising pathways of finding adult stem cells that could be used for treatment of Parkinson's. I mean, that there's a pathway, potential pathway available, and that was really important, you know, from a Catholic ethic, to be able to find that source and use it to treat so many different kinds of diseases.
Absolutely.
Yeah. So you mentioned a bit that you've been doing research at Molloy. So you have students helping you with this research. We're talking about that too?
Absolutely. I have students here with us today.
Are they here in the audience?
Yes,
they are? Okay.
Several groups of students.
That's
students
great.
from previous years, students from current years. Most definitely, we do a lot of work with.
So what are they doing exactly? Like what kinds of things
Okay,
they do?
so I just met with one of my groups before coming here today, and we are looking at a way to use our biotechnology to knock down some of these markers, like make them not on the cell anymore, using siRNA, we call it. And so they don't express the marker anymore.
This
and
is a genetic process.
Yes, it's you know, involved with the genes. And then we're looking at, with and without this marker, what changes, what activity changes, what changes when they interface with the Pac-Man or the macrophage cell. Does it?
Yeah, when you said Pac-Man, I thought, I was back going, macrophage.
Yeah, of course.
That was a good description. I love that.
Yeah.
So yeah, so I understand that. So you're doing, so have you had to do the DNA sequencing and those things on these different cells, so you know which part you're modifying?
Well,
or is it
we
not
have
that
to know what
precise?
the sequence is. We have to know what the messenger RNA is,
Right,
really.
okay.
The DNA, the message is
Right.
what is going to
Okay, that
become
makes
the
sense.
protein.
Right.
So we're knocking down the transcription of that gene, the message that the nucleus is giving through the messenger RNA, and that is what we're trying to knock out, because that is what then directs the
cell
production of the protein.
Yeah, what to do, right, exactly. So how did you end up getting interested in this as your research area?
I have been doing this research for at least 20 years. I started at Winthrop.
What were you doing your research on when you started? Were you working with stem cells, or were you?
I was working with mesenchymal stem cells, with these adult stem cells, directed toward bone biology.
I see.
Yeah, bone biology, bone disease, working at Winthrop University Hospital. And that was my area. We would isolate them and look at the differentiation process, or the process of them becoming bone cells.
I sometimes don't think people appreciate how scientific research takes place, because you said you've been working with this for 20 years, so with a similar cell line? How long have you been working with the same cell line?
Well, I've been isolating these cells from the bone marrow. We use animal models, of course, because that is you know, the process to start with an animal model. So how long have I been isolating these cells? For about 20 years, but different lines of research with them.
Right, okay.
Different
That's
lines of research. Bone biology I started with, and then I wanted to make a bridge between bone biology and cardiovascular biology. And then the research has morphed over the years along with the trends.
How do you get your inspiration as to how you, because let me mention something first. So when you do this research, how many hours have you invested in this research, do you think, over those 20 years, Jodi?
Oh, thousands of
Oh, yeah.
hours.
So, I think if you want to be a research scientist, I think you have to understand that you can have a great idea and concept, but boy, a lot of hours goes into doing the science, right?
It's persistence.
It's persistence.
It's just the word research. Research. You're always redoing results that we got today. We did it twice. We looked at the results. They didn't quite match. We have to go back and
figure out why.
We have to go back. You have to be a very persistent personality. You can't be stopped by problems. You have to just continue.
Yeah. And so, where does your inspiration come from in terms of how you're going to decide you want to spend that amount of time on a particular direction?
It's just naturally, I've always naturally asked questions, and I want to find the answers, and I know I can, just continually asking those questions. Wouldn't it be nice if we could just find an adult stem cell and make, almost make it very unique to everybody's disease, kind of custom-made stem cell treatment is
well, when
kind
you
of
think
a goal.
about so many of the debilitating chronic diseases we have, and you were mentioning them, and we see significant numbers of those as being autoimmune diseases now too, where the body is destroying its own tissue or with the inflammation and so forth. You were describing that earlier. But so many of these are chronic situations we find ourselves in. So the treatments we have are surgical treatments, or we reduce the pain, the inflammation to some degree, which might reduce some of the pain and symptoms, but we can't really solve it.
No, we haven't. We haven't solved it. We use steroids to treat
Absolutely.
the inflammation. We have new biologics now that prevent some of these inflammatory responses. The production of the inflammatory cytokines are targeted now. But what happens with that is when you are targeting those inflammatory cytokines, as I said, those are also a natural defense. So what you are doing by stopping them and treating the disease, you are also suppressing the person's immune system.
For other things.
For other things.
Right.
Absolutely.
That's hard.
So that's one of the problems.
Yeah, so you wouldn't want to do that lightly or early in the when the
Precisely.
diagnosis occurs. You'd probably do that more if the symptoms are really more severe, right?
Absolutely.
Yeah, and I suppose that's part of the thing too, that most of what we have available we don't arrive at early in the diagnosis either with the treatment of many of these things, I would imagine too. With what we have available now. You're not going to get the surgery. Well, you won't get it approved, that's for sure. You kind of know how that works. You won't get it approved until it's really far along anyway. So you talked about there being over 400 clinical trials going on right now?
Yes, through the NIH.
And so, so you think we're on the verge of some real excitement?
We are. We're on the verge of some exciting new treatments being developed. But I just want to make, caution that if you are going to participate, make sure it's an NIH trial, that it's a trial that is
sanctioned.
Sanctioned, yes.
Yeah. One of the things we talk about at Molloy is, how do you do research? And there are good ways to do research and not so good ways of doing research. And you want to make sure that all those protocols have been looked at.
Yes, and that you're informed. You make sure that you're well informed.
So clinical trials are one thing, but by the time it will get to market for the vast majority of Americans, that takes a little longer.
It does, because then it goes through FDA and that. But some of these trials are in stage 3. Which means? Which means they're very close to that.
So that's exciting.
Yes.
Yeah. Well, I appreciate you coming and talking about
Thank you.
Stem cell research and what it means and for individuals that have some of these chronic diseases. And it's exciting to know that we're making progress and we're able to do it in a really ethical way. And Jodi, I appreciate you coming and sharing that. And we appreciate you coming and joining us on the Public Square 2.0. I'm Drew Bogner, president of Molloy College, and I hope to see you again on another episode of the Public Square 2.0. Thanks for being with us.